Okay. All right, if you'll get out your notes, page 25, and on the back of that, you should have some extra space there. And if you have not already drawn your enzyme picture, I'm going to kind of guide you through that and help you know what to draw. Mainly this picture here sums up most of it. There's going to be an additional part that we're going to add, but this picture shows most of what's going on with the enzyme reactions. Now, enzymes, what they do is they accelerate the rate that reactions take place. Like we talked about yesterday, if you wait for, these things would happen naturally, but if you wait for them to happen naturally, things would not progress very fast, and a lot of your metabolism would not occur. So we need the enzymes to speed them up, and oftentimes at the rate of thousands of interactions per second. So this is an example of sucrose being broken down into glucose and fructose. So what you need to draw is this enzyme right here. Well, whoops. Hold on. Let me go back. There we go. Okay. All right, so you need to draw this enzyme right here. Okay. Label the purple thing there is your enzyme. And this entire thing is called an enzyme substrate complex. Okay, now you may want to use, if you want to use colored pencils to kind of differentiate what you got on your, for your enzyme and your substrates, that's fine too. Uh-huh. Okay, and so you're labeling it as the enzyme substrate complex. Yes, this entire thing is that. The purple itself is the enzyme. And then the yellow is the substrate. And in this case, it's sucrose is what the substrate is. And it could be, anything. But that's just the example that's given here.
Are we going to draw like all of them or just that one?
Draw this one, and then I'll show you how to go from there.
Like start over here.
Yeah.
I got a bunch of stuff drawn.
Okay, that's awesome. That's awesome. You're good. All right, now the place that the substrate joins to your enzyme is what is called the active site. So the active site is actually right here. It is where the substrate is going to lock in, like it mentioned, the lock and key. All right, so that is the place right there, and you'll see it here. It's labeled for you. Here you can see that it shows you the substrate fits right into there. And then if you'll draw this arrow here, and what we're doing, in this case, it's hydrolysis. We're introducing water so that we can break those apart. The enzyme uses water to break them apart. And then if you'll draw this enzyme, okay, and then show these are your products. It breaks it down into glucose and fructose. You don't have to. Okay? So what enzymes do is they can either take two substrates and put them together, or they can take a substrate and take it apart. Okay? So either thing can happen. They're just speeding up the rate that these chemical reactions are happening. Okay? I do want you to look at this this chart. what the enzyme actually does is it decreases the amount of activation energy that you need for a reaction to take place. So that is what causes it to speed up. It accelerates the molecules and causes the molecules to go faster so that they are more likely to come in contact with each other. Okay? So whereas it took, you can see the black line, it shows how much energy the EA is activation energy. So without the enzyme, it took a lot longer, a lot more energy, and a lot more time for the reaction to take place, whereas the red line shows that with the enzymes introduced, it doesn't take quite as long, okay? So it speeds up the reaction by lowering the activation energy. Now, I want to draw your attention to number 5 on your outline. It says that factors that can affect enzymes' ability to work optimally, so at their best. Temperature. If it's really cold things don't move as quickly, so that can influence how fast your molecules are able to move. Too much heat can denature the protein. Remember, that will cause it to unfold. Okay, so when you're considering that, let's see. They're showing the denaturing of the protein there. All right, so when you consider that, the enzymes speed up the rate. Heat speeds up the rate of reactions too. But it denatures the proteins in the process, so then they can't work. And remember, enzymes are proteins. So if it denatures them, then it does no good. So enzymes in and of themselves are able to speed up the process without heat doing that, because they'll cause more harm than good. So pH, or the POH, affects the ability of the enzymes to work too, because it messes up the hydrogen bonds. Salt concentration also will mess up the hydrogen bonds. So optimal conditions are going to be the things listed there for most human enzymes. Is your body temperature is right about normal, your pH is 6 to 8, and that creates an environment so that it's more stable. All right, so what I want you to do is I want you to use your manipulatives that I handed you. Okay, so you see in the active site that you've got your substrate and that it can work. But like I said, that's not always what happens. There are also things that are called inhibitors. And it mentions that at the bottom of page 25, and then it's going to talk about it at the top of page 26, okay? This blue structure here that you have in your manipulatives, and that is shown right here. This is a competitive inhibitor. And what that means is this is not the substrate, but it can fit into the active site to block the substrate from coming in. So let's say your inhibitor is present and my substrates come along, they can't get in there. Okay, so they can't work. Because there's a competitive inhibitor is competing for the active site. Okay? There are also things called non-competitive inhibitors. And that is what is pictured right here. Okay? They will attach to another place on the enzyme called the allosteric site. Now on your enzyme, if you'll notice somewhere, there is an opening on the opposite side. Y'all see that? Okay. It's not competing for the active site, but it will go in and lodge itself in the allosteric site, and it changes the shape of my active site. So now I can't fit my substrates in there because the shape changed. Okay? So I can't fit them in there anymore. Alright? So the allosteric site is the place that non-competitive inhibitors will go to turn off the enzyme. So, okay, so you got your allosteric sites often on the opposite place as the active site. Okay? So on your picture, I want you to add down at the bottom, I want you to add a little allosteric site. Just do you a little gap there. Go back in and make you a little gap. Okay, on your picture that you drew. Well, both, I guess, since it's showing that enzyme. The same enzyme. So you're going to put that as your allosteric site, where a non-competitive inhibitor would come and stop this from happening.
Why would they need allosteric?
Okay, that is a great question. Why would you ever want an allosteric site? Why would you want to have a place on you that would stop this from happening? Anybody have any idea?
So it don't reproduce
Okay, to either slow it down or stop it completely. This is a method that organisms have to control how much of something is produced. It is called negative feedback. Okay, so let me show you this. Okay, so what you've got here is, let's say that we want to make, in this case, in this picture, is isoleucine. Okay, so that's what we're making as our substrates. Okay. Well, let's say that we have made a bunch. We don't need any more. There is a problem with having too much. First of all, it uses up resources and energy that you could be using for something else, and you're wasting energy on making something you don't even need any more of because you have plenty. So sometimes it's because the extra isoleucine that you produced, actually the product will come and attach in that allosteric site to shut it off. Because it's saying, I've got enough, stop. Okay? And then it allows the isoleucine to be used up by the cell. And as it's used up by the cell, then this gets used up too. And it opens the active site up again to make more. Okay? So that's a good question. It's called negative feedback. And on the back, it talks about that in Roman numeral 8. It mentions negative feedback loop. And what that means is that you're making, and something's going to happen to slow that down or stop it so that you're not making too much, producing too much. Okay, does that make sense?
Yeah.
Okay. Okay, so one of the math problems that they can use with talking about enzymes and rates of chemical reactions is something called the Q10 temperature of, coefficient. All right, and what that means is that it's used to see what happens to the rate of a reaction if the temperature is raised by 10 degrees. Okay, so that's why it's Q10. If you will notice on your gold math sheet I gave you, the Q10 temperature coefficient is on the back underneath rate and growth. Okay, so it has Q10 equals, and then it's K2 divided by K1 with a coefficient of 10 over T2 minus T1. Okay, it looks really intimidating. It's really not bad. Okay, I promise. So if you're looking at this, like I said, it's the factor by which the rate, which is K is the rate of a reaction, increases for every 10-degree rise in the temperature, which is going to be T. All you need for this is 2 temperatures and 2 rates, okay? And the units have to match, okay? Now, when you are doing your answer will not be, it actually will not have units in it because it's more like a ratio. So, okay, so let me give you this. If you will get a piece of paper, and I do want you to know this because this is going to be another one of these math problems that was included on that math quiz. It's on the back. So this will be something that you'll be asked about too. All right, but it's really easy. All right, so let me go back here, and if you'll write down that Q10 is the factor by which the rate, which is K, of a reaction. So the rate that it increases for every 10-degree rise in temp, which is T.
What page are you going to? Or is this older notes?
This is not on your notes. It is on an extra sheet that they gave me. So it's not in that page of notes. It just goes with the enzymes because you're doing rate of reactions. So that's why they stuck it in right here. Okay, so the things that you're going to be given, here's your formula, and you do have that on you've got that on your math sheet, okay? The thing that is different maybe about this, the only thing that could be potentially confusing is that T2 is not necessarily the second time. That's what I thought the first time I ever saw Q10, was I thought it was the second time. It's not. T2 is whatever the highest temperature is. So no matter if that happens at the beginning of the reaction or at the end, you take the highest temperature, and that's going to be your T2. T1 then is going to be the lowest temp. And K2 is going to be the rate, whatever the rate was that goes along with T2. So T2, K2 go together. They're talking about the same part of the reaction. And then when I show you a problem, it'll make sense, okay, when we start plugging it in. And then K1 is going to be the rate when the temperature was the lowest. Okay, so it goes with T1. Okay, so I'm going to give you an example. I'm going to give you a sample problem, okay? So here's our practice problem. All right, we have a goldfish that's been put in a bowl of water. This water is 17 degrees Celsius. And it has a respiration rate of 110 OPMs. That's operculum movements per minute. That's the little openings on the side of their face. Okay, so that's its rate. It's opening at that much. After cooling the water with ice, the goldfish is placed in a bowl of water at 10 degrees Celsius. Okay, so it's going to be 10 degrees that time. And then it's going to be a respiration rate of 62 OPM. Okay, so what you got to do is basically plug this in to your formula. All right, it is not important here that you change your degrees to Kelvin. Okay, so you may leave those in Celsius. All right.
Is it the temperature coefficient thing?
It is. So it's going to be temperature coefficient Q10. It's hard for me to write on this little one. It's not showing it.
Yeah, I didn't know if it was supposed to work.
Yeah, I'm showing it on my thing, but it's not showing. Okay, so what is our K2? 110. Okay, so we've got 110 over 62. All right, and then our exponent is 10 over 17 minus 10. Okay, so if we take our 110 divided by 62, that gives us 170. Okay, so your rate is 110 over 62. Okay, and your exponent is 10 over 17 minus 10. All right, so if you figure that out, that'll be 1.77. Your exponent will be 1.43. You'll need a scientific calculator to do that, but that'll give you a Q10 answer of 2.26. And there's not a unit that goes with that. You'll be able to just leave it there. And for most biological systems, the answer will be somewhere between 2 and 3 because that rate is consistent with life.